Aloha everyone, and welcome to our lecture on blood. Today we'll be talking about the functions and properties of the blood within your body. So blood is a connective tissue. It's a liquid that connects all of the cells in your body. It's comprised of cells surrounded by a liquid matrix, which is known as blood plasma. So that plasma comprises 55% of your blood. The rest of your blood is comprised of cells, which is going to include red blood cells, white blood cells, and platelets. This blood has been centrifuged, which means spun around in a machine that separates things out by density. So what that means is that the red blood cells are going to be the most dense, the white blood cells a little bit less dense, and the plasma, which is just liquid, is going to float on top because it's less dense than the others. So the components in the blood that are cellular or formed elements are going to include red blood cells, which are anucleatic, that means they don't have nuclei, white blood cells, which are going to be involved in our immune response, and platelets, which are going to be involved in our clotting response. The plasma portion of the blood consists mainly of water, but also of proteins and many other solutes like ions and electrolytes. So if you were to look at your entire body weight, 92% of your body weight is comprised of other fluids and tissues that are not blood, and 8% of that is comprised of full blood. 8% is quite a lot, though, if you figure that their normal human weighs around 150 pounds. That's going to put you at about 12 pounds of whole blood. That blood, again, is split up into plasma. It's about 55%. And those formed elements that we just talked about, which are 45%, the formed elements are going to include things like platelets. Again, these are involved in your clotting response. Your white blood cells, which are involved in your immune response. Your red blood cells, which are going to be your oxygen delivery system. Let's look at these white blood cells for just a second, and don't worry, they're going to get their own slide later on in the lecture. The white blood cells are going to include neutrophils, that's 60 to 70% of them, lymphocytes, 20 to 25% of them, monocytes, eosinophils, and basophils, which comprise 5% or less each. In the plasma, we're mainly going to be looking at water as our primary component, but we also have a lot of proteins in there. Some of the proteins are going to include albumin, so you might hear that from like bovine serum albumin, for example, globulins, fibrinogen involved in the clotting response, and a bunch of other proteins as well. Some other solutes you might expect to encounter are going to include electrolytes, nutrients, gases, which really the whole primary reason that we have the circulatory system is so that we can have gas exchange that's going to work in concert with the respiratory system. Also different regulatory substances like hormones, etc., and then waste products which are on their way to be excreted. This here is what blood cells look like under a scanning electron micrograph. And just to let you know, anytime you see an electron micrograph, it's not a light microscope, so there's never any color. So everything here is pseudo-colored. Normally these are black and white. This is what a white blood cell looks like underneath the electron micrograph. This is the red blood cell, and this is the platelet. And you can see the red blood cell is dimpled, and that's because it doesn't have a nucleus. So it has a very unique shape, kind of like a donut that is not quite complete in the center. We also have a white blood cell. You see they have a lot of little sticky regions. Those are because they're going to be involved in the immune response, so they're going to be digesting anything they come in encounter with that might be considered a bad guy. Here we have platelets. You can see they look kind of funky with these really long legs, and those long legs actually are kind of like a chain of monkeys, and they're going to change shape if they need to have a clotting response occur. So in response to an injury or a cut, they're going to change shape, and that's going to mean that they're going to kind of coagulate together to be able to form a plaque wherever we have a site of injury. We'll talk about that later on as well. This here is a blood smear, so basically if you're looking at whole blood smeared out on a glass slide, what are you going to see? Well, mainly you're going to see these red blood cells. That's what these erythrocytes are going to look like here. Erythrocytes are another word for, fancy word for red blood cell. You also see platelets here, and white blood cells. We have a ton of different types of white blood cells. So this one is a neutrophil, this one is a monocyte. They're both going to be white blood cells, but they have slightly different functions. So they do have immune response functions, but they're going to have response to different invading pathogens. We'll talk about that later on in the lecture. So the function of blood is to transport things, right? Nutrients in, waste out. So it transports things like oxygen, carbon dioxide, different nutrients, hormones, heat, as well as waste products back out. It also regulates homeostasis of all of your bodily fluids. It regulates your pH, regulates your body temperature, and the content of water inside your cells. And it has clotting factors in there that are going to help regulate against excessive blood loss. And also it helps regulate against infections by using these white blood cells. So white blood cells, as I mentioned, are part of your immune response, which are going to help attack any foreign invaders. So if you were to look at the blood plasma as a whole, we have about 91% of that is going to be comprised of water, which is just the liquid portion of the blood. But water has some unique properties in that it's going to help really absorb heat, so it's going to hold heat. It's also going to be a really nice solvent, so almost everything dissolves in water. We have some things that are not going to dissolve in water, right? Things that are lipid-based, and they might have to be carried separately. But in general, most things are going to be held in a water medium. We also are going to have some plasma proteins. These guys are going to be produced by the liver, responsible for all sorts of things. Remember, proteins basically are the workers of the cell and of the body, but these are going to be responsible for things like the osmotic pressure, so the amount of water pressure inside the blood plasma. Also contribute to things like blood viscosity. There's a lot of transport hormones in there that are going to be moving around the circulation, also fatty acids and calcium. A lot of these proteins are going to be involved in maintaining homeostasis, so maintaining blood pH, for example, or osmotic pressure. So albumins, globulins, and fibrinogen are all plasma proteins that you're going to find that are going to play roles in things like blood clotting or regulating blood pH or blood pressure. Other things you might find, I apologize, are different electrolytes, nutrients, and gases. Some electrolytes include inorganic salts, so things that are positively charged or negatively charged. So if they're positively charged, they're called cations. If they're negatively charged, they're called anions. And cations include things like sodium, calcium, magnesium, potassium. Negatively charged things include chloride, sulfuric acid, etc., bicarbonates. And these are all going to be responsible for maintaining osmotic pressure as well as maintaining blood pH in some cases, like in this bicarbonate system, for example. So the nutrients we're going to see are going to be things that are going to be coming in through the products of digestion. So the digestive system and the circulatory system are in tight conjunction, and so anything that's going to be picked up from the intestinal walls, for example, and then end up in the circulation is going to be involved in distribution around the rest of the body. So nutrients, for example, that play a role in cell functions and growth and development. Some other things you're going to find, in fact, this is the first thing you think of when you think of blood supply, right? Oxygen and carbon dioxide, as well as nitrogen, but gases that are going to be involved in cellular functions, involved in regulation of blood pH, etc. And we're not really sure exactly what nitrogen is doing, but we do know that we have nitrogen gases in our system. Some other regulatory substances, including enzymes, hormones, and vitamins. Enzymes are going to catalyze chemical reactions, and this is such a broad spectrum. This basically is just saying they do things, right? They do work within the cell. And they have specific reactions that they each regulate. So, and one enzyme has one specific function. We also have hormones that regulate things like metabolism, growth, and development. Vitamins, which are basically cofactors, so they work alongside the enzymes to make sure that reactions happen. We have a ton of waste products as well, things that you're going to see excreted. Generally, you're going to see them excreted either in your urine or in your feces or out through the respiratory system in cases of like carbon dioxide gas. So waste products like urea, uric acid, creatine, bilirubin, and ammonia are basically going to be things that are byproducts that are going to be excreted out. It says organs of excretion, but all of these are going to go out. Most of these are going to go out through the urinary system. All right, so how are blood cells made? So some blood cells are able to survive for a much longer time than other blood cells. So for example, lymphocytes are able to live for years, and other blood cells only live for hours or weeks. And we are constantly need a steady amount of certain amount of blood cells. So for example, red blood cells, we need a steady amount of. Platelets, we need a steady amount of. But the amount of white blood cells in our system basically depends on what kind of invaders we have. So a country that is at war, for example, needs more soldiers than a country that is at peace. So when you are under attack, if your immune system is actually under attack by some sort of infection, the white blood cell count is going to increase. But the process of producing white blood cells and red blood cells is hemopoiesis or hematopoiesis, and that's basically how we're going to make the blood cells. Poiesis just means to create, and hemato is referring to blood supply. Pluripotent stem cells are going to be the cells that are going to create these cells. And pluripotent stem cells are basically going to be cells that are going to be able to differentiate into multiple different things, but not everything. So something like an embryonic stem cell is what we call totipotent, or it has total potential, and it can become anything in the entire path of cell lines. Pluripotent means that it already has kind of picked its path, and it can become one of many different things already within a particular family. So for example, they can become any different type of blood cell, but they're never going to become a nervous system cell, for example. So a pluripotent stem cell is what is going to be a precursor cell that differentiates into the different types of blood cells. And so what does this look like? This is a huge graph, and there's a lot to take in. So let me just start by telling you I'm never going to ask you specific steps in the pathway of any of the particular types of the leukocytes or erythrocytes. But I want you to understand that there are key steps in these pathways. So let me give you an analogy here. I like to consider stem cells to be something like a kindergartner. So, for example, the totipotent stem cells that I just talked about, the embryonic stem cells, that's a kindergartner who is able to undergo any different pathway. They can become anything they want when they grow up. But as you start to go through the system, you graduate high school, you get into college, right? You start to have a major, and you are probably going to do something within a specific field. And that's what this pluripotent stem cell is, right? He just graduated college. And if you say, for example, he has a biology degree, he could work either in academia or in the research industry, or he could do a lot of different things with that degree. But one thing he's probably not going to do is become a carpenter, right? So this pluripotent stem cell is able to enter into one of many different pathways, but they're all going to be in the blood cell lineage. And one of the first decisions that it's going to have to make is whether or not it's going to enter into the myeloid stem cell line or the lymphoid stem cell line, right? And if it's going in the myeloid stem cell line, then it has another differentiating decision to make. And it's either going to go down the erythrocyte pathway, the megakaryocyte pathway, or in this case, it's going to become the mast cells, right? It also can become a granulocyte macrophage. Now, if it heads down this erythrocyte pathway, it becomes a proerythroblast. So both the word pro and the word blast indicate that it's a precursor, right? Pro usually means that it's going to have a cleavage event or something that happens that makes it whatever the next thing is, right? So it's going to be a proerythroblast. It's a precursor. It still has a nucleus, but as I just mentioned previously, erythrocytes don't. So red blood cells don't have a nucleus. So the nucleus is going to have to get ejected out of it, and then that becomes what's called a reticulocyte, which is kind of an intermediary molecule, and then eventually goes down the pathway to become a erythrocyte. Now, this is a differentiated cell. All of these are differentiated cells. These pathways do not work backwards. So once you become a red blood cell, you stay a red blood cell. But the red blood cell came from the same lineage, the same pluripotent stem cell lineage at the very beginning that all of the rest of these cells came from. All right, so I'm not going to walk you down each of the pathways. So basically, steps are made, right? This step goes either myeloid line, that goes to lymphoid line. If it goes to lymphoid line, then it can go into T lymphoblast, B lymphoblast, or NK lymphoblast. What does that stand for? Well, if you were here for our immunity lecture, you know that would be T cell, B cell, and natural killer cells. So, right, so the T lymphoblast then become the T lymphocytes. So the T cells, B lymphoblast become the B lymphocytes, or the B cells, and the NK lymphoblast become the natural killer cells. And as you can see, just like what I mentioned previously, the more specialized a cell becomes, the less of the options it has available to it. So as it starts going down this pathway, it really only has one choice after a certain point. So it reaches a point of no return where it has entered into differentiation and development and can only become that particular cell. Right, again, I'm not going to walk you through all of these, but one thing that I do want to draw your attention to here is this CFU MEG, which is basically a colony forming unit. If you see CFU, and that's just a way by which they differentiate which of these cells lines it's going to turn into by using bacteria. So you can ignore the CFU, but what you want to look at afterwards, MEG is for megakaryocyte, E is for erythrocyte, GM for granulocyte macrophage. Now, this megakaryocyte becomes a megakaryoblast, right? Oh, sorry, becomes a megakaryoblast, becomes a megakaryocyte. I apologize. It's entering into the megakaryocyte cell line. And the megakaryocyte itself is going to break down to become platelets, and platelets are going to be what's involved in blood clotting. So platelets themselves aren't cells, right? They're pieces of cells or fragments of this megakaryocyte that just constantly creates a lot of cell products, basically. And so they have a much smaller lifespan than some of the other cells because they aren't actually cells. They're kind of like an intermediate, a cell product. All right, so the red blood cells, or the erythrocytes, are going to carry oxygen. And they do that by using a protein called hemoglobin. Hemoglobin is basically a bus that has seats on it, and the seats can be occupied either by oxygen or by carbon dioxide. So they carry oxygen to all cells, and 23% of the carbon dioxide that's carried through the bloodstream is carried on the red blood cells. But some of that carbon dioxide that's carried through the bloodstream is going to be solubilized in the blood plasma as well, and we'll talk about that in a different slide. Now, each of these hemoglobin molecules that we talked about has an iron ion, and that iron ion is going to be the seat. So we have 4 seats and combined to 4 oxygen molecules, or they can also carry carbon dioxide molecules if we don't have enough oxygen in circulation, which is how we get our carbon dioxide out after we drop off our oxygen. As I mentioned, the red blood cells don't have a nucleus, so they're anucleatic. They don't have any other organelles. And they have an odd biconcave disc shape, kind of like a torus almost. And the lack of the nucleus and this unique shape allow the cells to carry oxygen very efficiently. So So what does that look like? So this is what the red blood cell looks like. As I mentioned, it's kind of like got a dimple in it, and if you did a section view, you can see it kind of looks almost like an 8. And inside here we have hemoglobin. So this is the hemoglobin molecule. It's a tetramer. So when I say tetramer, I mean it's made of 4 different parts, part 1, 2, 3, and 4, and you'll note that these 2 are the same and those 2 are the same. So we have alpha and we have beta hemoglobin. We also have gamma hemoglobin, which is found in the fetus, and that's actually going to account for the higher affinity of the fetal hemoglobin molecule for oxygen than the normal molecule for oxygen. And that's going to be very important because if you can picture that the maternal blood is already circulated through mom's body, so it's already delivered some oxygen. In order for the fetus to be able to pull that oxygen away from the maternal blood and onto the fetal hemoglobin, it's going to have a higher affinity than the maternal hemoglobin does. So gamma hemoglobin is a specialized type of hemoglobin that's only found in fetal circulation and has a higher affinity for oxygen than the alpha or beta subunits. But I digress. This is what the heme looks like. The heme is shown here in red, so this is the iron group here. The whole section is the heme group. And this is going to be basically the seat where either oxygen or carbon dioxide is going to sit down. Hemoglobin has other roles as well in the blood supply. It's involved in regulating blood flow and also blood pressure. And it does that by releasing nitric oxide. Nitric oxide helps vasodilate. So vasodilate basically just means that the radius of the tube is going to get larger, so dilation, getting larger. And that's going to allow more blood to go through in a shorter period of time, and that's going to help enhance the delivery of oxygen to the tissues. Inside the red blood cells, we also have something called carbonic anhydrase. And carbonic anhydrase is responsible for the conversion of carbon dioxide into water and carbonic acid. And that's going to be how approximately 70%, that says OC, but it should say of, approximately 70% of carbon dioxide is carried in the blood plasma in this carbonic acid. And what's interesting is it's also a buffer, which means that it's also going to help maintain our blood pH, which is very important because just like any homeostatic condition, if it wavers too far in either direction, either too acidic or too basic, we can end up with problems. Now, red blood cells are pretty shortly lived. They only live for approximately 120 days, and then eventually they make their way through to the spleen and the liver where they're degraded. So they're taken out of circulation by the spleen and by the liver. And these organs are going to break those cells down and recycle them and reuse their components to be able to create new red blood cells. Now, don't let me give you the idea that red blood cells are created in the liver. They are not. They are created in the bone marrow, all right, just to be clear. Oh, look at this. So red blood cells are created. I'm going to start at 8 and then we'll go to 1, all right? So they're actually going to be created, so erythropoiesis starts in the red bone marrow of the bone, and then it's going to end up released into the blood. Goes into circulation for about 120 days where it's doing its job, picking up oxygen from the lungs, dropping off oxygen to the tissues, picking up carbon dioxide from the tissues, dropping it back off of the lungs, lather, rinse, repeat, until it starts to get old and break down and decay. All right, so then it's going to make it to either the spleen or the liver or the red bone marrow. So there are 3 places where it can end up dying, basically going to die. And it's going to undergo digestion inside this macrophage. So we have macrophages as a part of the immune system. They're found pretty much all over the body, but they're found in higher concentrations in the spleen, the liver, the red bone marrow, and other types of lymphatic tissue. But these macrophages are going to ingest the red blood cell by phagocytosis, and then it's going to get broken down into 2 different sections. The globin section, which is going to be reused. So that's going to be turned into amino acids and reused for protein synthesis. So that's going to be recycled. The heme is going to have 2 different things happen. So it can either get converted into biliverdin, which eventually gets converted into bilirubin and makes its way to the liver, or it can have this turn into Fe3+, right, with the addition of transferrin, which makes ferritin, right? And ferritin is also going to make its way to the liver, and then it's going to end up with that Fe transferrin again, which is going to end up heading back into the bone marrow, where it's going to be used, right, that Fe3+ is going to be used in the addition of globin and vitamin B12 and erythropoietin, which is a specialized molecule which helps create red blood cells, so it helps with erythropoiesis. Basically, my point is that this Fe3+ is going to get recycled back to the bone and help drive the creation of new red blood cells. Okay, so we've accounted for 2 different pathways, right? Globin is going to go to amino acids, get reused for protein synthesis. Heme can do 2 things. It can do the ferrin pathway that we just talked about, right? It gets turned into ferritinin and then ferrin transferrin and then back into the bone marrow where it's used to drive erythropoiesis of new red blood cells. So this last one here is what happens to the biliverdin. It gets turned into bilirubin, which then ends up in the liver. It's eventually going to get secreted out of the liver and end up into the small intestines. Okay, while it's in the small intestines, it's going to, here's the bilirubin in the small intestines, bacteria are going to convert it into something called urobilinogen, and that can get sent to the kidney in the form of urobilin to be secreted in the urine. And the byproduct of that is something called stercobilin, which is just going to be secreted in the feces. So basically this bilirubin and the biliverdin ends up going into the liver and then gets secreted out in the waste, either through the small intestines, well, by way of the small intestines, into one pathway that goes out through the urine, and then another pathway, the stercobilin pathway, that goes out into the feces. Okay, so again, some of the definitions that we just talked about, erythropoiesis is basically when we are creating red blood cells. That happens in the red bone marrow, so inside the bone tissue. Reticulocytes are the immature red blood cells, the babies. They're going to enter circulation. Then about 2 days later, they're mature, and then they're going to go into circulation for about 120 days and then enter into that degradation pathway we just talked about. One of the proteins that we saw, this is erythropoietin. We saw it here, creating or driving erythropoiesis, or the creation of red blood cells in the red bone marrow. So erythropoietin is going to be a hormone, and it's actually released by the kidneys. And it's released in response to a low oxygen concentration. That's called hypoxia. So if we have low oxygen concentration, we release this erythropoietin. That's going to create more erythrocytes, which means that we have more red blood cells, which are going to be carrying more oxygen. So that's hopefully going to help restore conditions to a higher level or normal level of oxygen concentration. So what does that look like? Okay, so anytime that we're looking at one of these pathways, we are looking at a disruption of homeostasis, which disrupts whatever that controlled condition is. And then the body's attempt to restore homeostasis. So in this case, we're disrupting homeostasis by decreasing oxygen delivery to the kidney and other tissues, which leads to these receptors in the kidney cells detecting low oxygen levels and increasing the amount of that protein called erythropoietin that we just talked about being secreted into the bloodstream. Erythropoietin is going to end up going into the red bone marrow, where the prourethroblast are going to mature more quickly into reticulocytes. So we're basically taking those stem cells that we talked about, those precursor molecules, the pluripotent stem cells, and using them to drive the erythrocyte pathway instead of some of the other pathways that perhaps it could be driven, right? And that's going to lead to the creation of more reticulocytes, remember that's a precursor to the red blood cells, entering the circulating blood. And in approximately 2 days' time, we're going to end up with a larger number of red blood cells in circulation, because remember the reticulocytes are going to take a day or two to mature, but they are still going to be able to carry some oxygen in the early stages, so they're still going to be helpful to get those early reticulocytes into your blood supply. And that's going to increase the oxygen delivery to the tissues. Eventually we're going to return our homeostatic conditions when our oxygen delivery returns to normal to the kidneys, which means that we are going to then cut off this pathway because we've gotten back to our controlled conditions, so we've returned to homeostasis. So I've talked about red blood cells. Now I'm going to talk about white blood cells. There's a whole lot of different types of white blood cells. White blood cells are called leukocytes. White blood cells are nucleated. That means they do have a nucleus, they do have organelles, but they don't have hemoglobin and they don't carry oxygen. They're not responsible for gas exchange at all. And they're classified into two main categories. And these categories are kind of archaic. Basically, they're classified into whether or not they end up with little dots that appear when they're stained. So either they're granular, meaning that they end up with little vesicles that appear when we're stained, or they're agranular, meaning they don't contain those granules, right? We have 3 different types of granular leukocytes. Those are neutrophils, eosinophils, and basophils. And we have 2 different types of agranular leukocytes, lymphocytes, and monocytes. And they look kind of like this. So when we say granular, that's going to be these guys here. See these little granular dots? Right. Again, neutrophil, eosinophil, basophil. These 2 are what are considered agranular. That's lymphocyte and monocyte. Now, white blood cells have a really long lifespan. Actually, they could live for several months. Some of them live for years. And their main function is immune response. So combating microbes that have invaded your body. So during this invasion, white blood cells generally are going to leave the bloodstream and collect at wherever the invasion site is. This is called emigration or diapedesis. It looks kind of like this. So this here is the capillary, right? Here's the epithelial cells that are lining that capillary. Endothelial cells that are lining that capillary. And you can see there's a little bit of space in between. And in order for these guys to squeeze on out, they have to do some binding. So this is a neutrophil. The neutrophil is going to be rolling along until it reaches a region of the endothelial cell that has selectins. The integrins are going to be on the surface of the cell. The integrins bind to the selectins, and when they do so, then we can have the process of emigration, right? Or as I mentioned, diapedesis. So they're going to squeeze through those endothelial cells, and then they're going to end up at the site of an infection. And one of the ways that you can tell if someone is fighting an infection or an inflammation is by checking the levels of the white blood cells in their body. In general, if they have elevated levels of white blood cells, they are probably battling an infection or some sort of inflammation at the very least. A low white blood cell count might indicate no infection, but it could develop in the presence of infection due to several different causes. One might be like an HIV infection, for example. So you still want to check out all of the different cell counts to determine if there was a problem, something called a differential white blood cell count. And what that means is you're going to be looking at the ratio of all the different types of white blood cells in the body to make sure that they are in normal conditions or normal levels. So as I mentioned, these are the different types of white blood cells: neutrophils, lymphocytes, monocytes, eosinophils, and basophils. Now, if we have a high count, that would be indicated in this column. If we had a low count of whatever the cell is, that would be indicated in this column. So a high count of neutrophils would happen if we had something like a bacterial infection or a burn or inflammation or stress response. And if we had a low amount of neutrophils, regular to a normal individual, we could indicate that we had something like a radiation exposure, which might kill off the precursor cells, drug toxicity, which might have killed off these cells as well, or something like systemic lupus, which is going to lead to something called erythematosus, and that is an effect of the body having a problem differentiating between self and non-self, and so it attacks itself as though it is non-self, which means that it attacks its own tissues as foreign invaders, and that can obviously be a problem because it makes you very sick. Lymphocytes, if we have too many of them, or a very high count of them, that might indicate an infection, but in this case it would indicate a viral infection. So see here, neutrophils higher in bacterial infection, lymphocytes higher in viral infection. So each of these play their own role in the immune response. We can also end up with a high count in certain types of leukemias, or these are cancers, right? Blood cancers. And also infectious mononucleosis, right? So mono, the kissing disease. If we end up with a low count, that might indicate that we've been fighting the illness for a very long time, and these guys just keep dying off faster than we're able to create them. So something like a prolonged illness, or also an HIV infection, can do this. Anything that can cause immunosuppression can also do this, or treatment with something like cortisol, which is used to help fight off inflammatory diseases. So hopefully you can help stave off that immune response, which means that you'd be lessening the amount of lymphocytes available. If we have a high amount of monocytes, that can indicate a viral infection or a fungal infection. We see this with tuberculosis, also with some leukemias, other chronic diseases. If we have a low count, that might mean that we have a suppression of our bone marrow, or cortisol treatment again. Eosinophils indicate that we probably have an allergic reaction or a parasitic infection. So again, each one of these has their own specific pathway that they attack, their own foreign invader that they attack, or that we might have an autoimmune disease. If we have a low count, we might be a drug toxicity, so we've kind of killed them off perhaps, or acute allergic reactions as opposed to a longer-term allergic reaction, right? So it's an immediate allergic response. High count of basophils might indicate an allergic reaction, leukemia, cancer, hypothyroidism. Low count might be, here's the first time we see this, but pregnancy is going to help suppress the immune system, because literally a pregnant woman is carrying non-self inside her body, and so she doesn't want, in this case, we don't want to kill off the non-self, right? We want to nurture that pregnancy. And so the immune response of a pregnant female is drastically lowered in response to pregnancy, because they want to make sure that the non-self fetus isn't attacked by the immune system. Also in response to something like ovulation, which might lead to pregnancy, or stress, or hypothyroidism. Okay, let's talk about platelets for a second. Remember, they came from those megakaryocytes. Megakaryocytes splinter into thousands of these fragments, and those fragments become platelets. The platelets have vesicles in them. They do not have a nucleus, and they're involved in clotting the blood. So they're involved in the blood clotting response under the influence of a specific hormone, thrombopoietin. We can make more of these platelets from the hematopoietic stem cells. So just like anything else, we have a reservoir that we have at a normal level, and then we can upregulate if needed if we end up with a response that's going to need a lot of clotting, for example. Platelets are going to be short-lived. They survive only 5 to 9 days, and they're only involved in the clotting response, really. So this is a summary of what's called formed elements. So formed elements are going to include platelets, but if we're looking at cells, we're not going to include platelets. So just to be clear, formed elements are going to include the erythrocytes, or red blood cells, going to include granular and agranular leukocytes, including neutrophils, eosinophils, basophils. Also going to include platelets. So sometimes we can have problems with our red bone marrow, right? For example, bone cancer. And in this case, we might opt for something like a bone marrow transplant, where we replace the cancerous bone marrow with red bone marrow from a normal patient. And it's collected from the donor, and it's usually collected by a very long needle that goes through the hip bone, through the iliac crest. We also have the ability to freeze down stem cells. So if we can collect stem cells from an infant immediately after birth, so we can take that umbilical cord and freeze those cells down. And then we have the ability, perhaps later on in that individual's lifetime, to be able to use those cells to be able to fight off diseases. And they obviously have advantages over transplanted cells because they're going to be the body's own cells. So instead of having to then take immunorejection drugs or help maintain something that keeps you from being immunocompromised, you don't have to fight non-self because the treatment, the course of treatment that you're using is actually from cells that are from the individual who's affected. So there are actually self cells. Okay, so what happens when we get cut? We just talked about platelets being involved in the clotting response. So how do we stop the bleeding? So the word hemostasis means stop bleeding, and it involves 3 major steps in the process. The first is the vascular spasm. So the vasculature actually is going to change in response to a cut. The platelet plug formation, and then the actual act of clotting, which is called coagulation. So this is what happens during hemostasis. So just to kind of orient you here we have the red blood cells and the platelets. These are both considered form factors, but only this is considered a cell. These are collagen fibers that are going to line here, and we have a damaged endothelial layer here, right? So it's the damage. When we have these platelets enacting with that damaged region, they're going to liberate ADP, serotonin, and thromboxane A2. That's called the platelet release reaction, and that's going to be the very first step in the platelet clotting response, which is going to form that platelet plug. So basically what happens is we go from a regularly normal shape, one that wouldn't interfere with blood flow, right, to one that's going to be very pointy and kind of sharp, and they're going to cause these guys to aggregate together into a platelet plug. After that happens, the very last stage is we're going to be putting in these fibrin threads. And then those fibrin threads are going to tighten down, which are going to trap the red blood cells, but also the platelets. So these guys are little platelets here that are involved in forming that platelet plug. These are fibrin threads that are starting to go across this region. It's going to ensnare some red blood cells, and unfortunately they're just going to have to be kind of casualties to the byproducts to this. Then as this clot starts to tighten down, the fibrin threads are going to tighten further and further and further until they've wrapped entirely around that, and this is going to cause an actual clot. Alright, so blood clotting involves several different coagulation factors or clotting factors. They're identified by Roman numerals or divided into 3 stages. We have the extrinsic pathway, the extrinsic pathway, and the common pathway. So the extrinsic and the intrinsic pathways are both going to lead to the common pathway. So you can see we have an intrinsic pathway and an intrinsic and extrinsic pathway. So the extrinsic pathway would be if we have tissue trauma, right, that happens kind of to the outside. An intrinsic pathway would be if we have blood trauma. So trauma happens on the inside or the inside of the endothelial layer. Okay, so if we have tissue trauma, we have tissue factor that's going to bind, that's going to bring in calcium. You'll notice this pathway also brings in calcium. So calcium is going to interact with activated X and then also with 5, so activated 10 and also with 5. And those guys are going to come together to meet prothrombinase. Prothrombinase is going to start the common pathway. So I'm going to halt here and I'm going to go back up to the intrinsic pathway. Now remember, the extrinsic and the intrinsic pathways are going to lead to the same result, right? To this prothrombinase, which is going to enter into the common pathway. But it's going to do it by a little bit of a different way. So if we have trauma or damaged endothelial cells, we have exposed collagen fibers, which aren't normally exposed. So once those guys are exposed, then the platelets are going to be binding to that region. They're going to end up injured, right, or activated. And that activated platelet is going to lead to the creation of these platelet phospholipids, which is going to then combine with activated X. And at the same time, this blood trauma damaged platelets is going to start the activation of 7. Okay, so activated 7 and activated, when I say X, I mean activated 10. So activated 7 is going to interact with calcium and then reach activated 10 to lead to prothrombinase and enter into the common pathway. Now you may ask me, what kind of questions could you possibly ask me about this? This seems kind of overwhelming, and it is. Really what I want you to understand is that there are three major pathways, one that occurs from the outside, one that occurs from the inside, and then either one of them lead to that common pathway, which ultimately lead to the same thing, which is basically the creation of fibrinogen, creating these loose fibrin threads, and the activation here of 8, strengthening those fibrin threads to make a nice tight plug. All right, so now I'm going to enter into, I just talked about A and B, both of them end up in prothrombinase. Now I'm going to enter into C, which is the common pathway. And you'll notice that all of this is calcium dependent. So calcium is going to be really important in many different stages of this response. All right, so prothrombinase is going to get cleaved, or is going to cleave. ACE is an enzyme. So prothrombinase is going to cleave prothrombin into thrombin. And then thrombin is going to lead to fibrinogen turning into these loose fibrin threads, and then with the activation of 8 is going to get turned into activated 8, right? Activated 9, I apologize. Activated 9 is going to turn into activated 9 to strengthen down those fibrin threads to tighten that fibrin plug. I apologize, it was kind of small on my screen there. All right, so these are all the different clotting factors. I am never going to ask you which clotting factor is involved in which step of the response, but you do need to know some of the major names, right? For example, fibrinogen and prothrombin, you've seen before, you're going to see again, you need to know. You'll definitely need to know it when we're talking later on things like circulation. We have thromboplastin, which is a tissue factor. The calcium ions you're going to see over and over and over again. And so just to kind of talk about some of the major factors that are involved here and where they come from, their source is going to come mainly from the liver, but also from the diet, and calcium comes from your bones, right? And this tells you which of the pathways that they're involved in. So you can see that the extrinsic pathway is 3 and 5, and then 3, 5, 10, and 7, and they're going to head into the common pathway. And then the intrinsic pathway is going to use 6, 7, 9, and so 8 and 9 and 11 and 12. Again, I'm not going to quiz you on which ones are going to be used at which step. I just want you to understand that the injury can come from internal or external and then lead to that common pathway, which eventually leads to tightening down of that clot, right? And once that clot consolidates or tightens down, that's going to help pull the edges of the damaged vessel together and hopefully make it waterproof or watertight, so nothing's going to be leaking out through there. Now, in order to do this, we're also going to be relying on something called vitamin K. Vitamin K is necessary for normal clot formation. It's not directly involved, but it's used to synthesize for the clotting factors that are directly involved. And when the clots are no longer wanted, they're dissolved by a fibrinolysin. So lysin meaning lysine, so breaking things open, so it breaks down fibrin, and that actually is called plasmin or fibrinolysin. All right, so let's talk about blood groups for a second here. So you may be A, you might be B, you might be AB, you might be O. Surely you've heard of that if you don't know exactly what you are. So let's talk about what that means. So we're characterized into blood groups based on the presence or absence of specific glycoproteins and glycolipid antigens on the surface of your red blood cells. So we have 24 different blood groups and over 100 different antigens. But they're genetically controlled, and so we end up with a very small amount of them that are actually going to be presented. They're going to vary among different populations, and they're going to be classified into A, B, AB, or O. And I want to clarify here that A and B are going to be what's called codominant. That means that if A and B are both there, they are both expressed. However, relative to O, A and B are both considered dominant. So if we have A and O, it's going to look like it's A. If we have B and O, it's going to look like it's B. So having the presence of A or B is going to mask O. And we'll talk about why in a second. So the only way to get an O phenotype would be if you had 2 copies of the O allele, because the O is actually the absence of the antigens. So you either have the presence of one, the presence of the other, or the presence of both, or the presence of neither. And in order to have the presence of neither, you can't have A or B there, right? Another antigen that we're going to talk about a little bit is called RH, and this is where we get our positive or a negative. So if you talk about somebody who's O positive or AB positive or B negative, that's when they're talking about that RH group. And that's present in almost everyone. Approximately 85% of humans are going to be positive for RH. Now, as I mentioned, this is going to be something that varies with your population group. So depending on what descent you arise from. So in a European American population, for example, we're going to have approximately 45% O. So about half are going to be O. However, in the Native American population, about 80% are O, and only 1% are AB, and that's because very small percent are B, so there's not very much prevalence of the B allele. So really the main dominating types are going to be the A and the O. Alright, so I don't expect you to memorize all of these, I just want you to understand that depending on your descent, you may be more or less likely to have particular blood types. Now over here is the Rh factor, and as we mentioned, it's approximately 85% in the European American descent. That gets even higher if you get into the African American descent, and once you hit the Asian and the Native American lineages, you're going to end up with 100% of the Rh positive. So almost everyone in the Korean, Japanese, and Chinese American lineages are going to be positive, whether they're A positive, O positive, AB positive, B positive, but they're going to be positive for the Rh factor. All right, let's step backwards to talk about A and B. So we're going to talk about it from two different directions. We're going to talk about it from the cell, so the red blood cell itself, and also from the perspective of the blood plasma. So blood plasma is going to have antibodies that are going to react with what we call antigens, or the sugars on the blood cells. But because we don't want to have an immune response against something that we carry, no one is going to have these agglutinins or antibodies against the glycoproteins that they carry. So what does that mean? I just told you a lot of stuff. Let's look at this slide here. All right, so this is the actual red blood cell, and this is the plasma. And I want you to kind of understand that these are equal and opposite. So if I have a type A blood, right, then I can do that by having a genotype of 2 A's or a genotype of AO, okay? Now, either way, that means that I have at least one allele that is expressing the A antigen or the A sugar. This is the glycoprotein. That means that I have the A sugar on my red blood cells. That means that I don't have the A antibody, because if I did, right, if I had the A antibody with the A antigen, we would have coagulation event and we would end up having death, right? It would not be good. This is why certain blood types can only get blood from certain blood types, right? You can't just randomly hook up a transfusion from a stranger on the street, because you may end up with a reaction whereby we present the A antigen with the A antibody and we end up with coagulation and clotting and death. All right, so let me get back to the type A individual. A type A individual expresses the A glycoprotein on the surface of the red blood cells, but does not have the antibody in their plasma. They do, however, have the antibody against B, because they don't have the B antigen. All right, and the reverse is true for type B. They have the B antigen on the surface of cells, the antibody against A, no antibody against B, right? If we are AB, that means that we are creating both A and B antigens. And this individual is heterozygous. That means they have gotten one allele that is A and one allele that is B. And we know for sure, this is the only individual, well, I guess this individual as well, that we know for sure the genotype based on the phenotype. So if the phenotype is AB, they are genotype AB. And that means that they express both A and B antigens, or A and B glycoproteins, on the surface of their red blood cells, which means they don't have either antibody in their plasma. Okay? Now, type O individuals, we know that this individual is O phenotypically, which means he is OO genotypically, which means that neither sugar is created. So they don't create sugars against A or B, or if either A or B sugar on their red blood cells, which means that they have antibodies against A and against B. This is why this individual is unable to receive blood from anybody that is not type O, because if we put this one in, we end up with reactions to both. If we put this one in, we end up with reactions to blue. This one, reactions to purple. So type O blood can only receive type O blood. However, it can donate to anyone, because it can go into here, this antibody won't react, this antibody won't react, there's no antibody here. So that's why this guy is considered the universal donor, and this one is considered the universal acceptor. It has no antibodies to anything. So you could add any of these blood types to AB, and they would survive. Okay, so this just summarizes what I just told you. Right? So if you have this is your antigen on the red blood cells, right? If you're blood type A, you have the blood sugar A. If you're blood type B, you have the blood sugar B. If you're blood type AB, you have both A and B sugars on your red blood cells. For O, you have neither A nor B sugar on your red blood cells. And if you're looking at the antibody, it's the equal and opposite to what you have in antigen. So if you have the A antigen, you have the B antibody. If you have the B antigen, you have the A antibody. If you have both, you have neither. If you have neither, you have both. Does that make sense? All right, so that means that you have selective compatibility with different donors. So, for example, an A individual is able to accept blood from either A or O, but not from B and not from AB. Why? Because these guys have B, and that B will react to the antibody in their plasma, and they will get sick, right? A B individual is able to take it from B or from O, but not from A or AB. Same thing because of this A, right? If the A sees the anti-A, you're going to have a reaction. AB is able to take it from A, B, AB, or O because it's able to accept it from anyone. Why? Because it has no antibodies in the plasma, none, so there's no incompatibility. However, O has antibodies to both A and B in the plasma and is only compatible with O, so it's incompatible with everyone else, including A, B, and AB. All right, so in order to determine a person's blood type, we have to type it and we have to cross-match it. That means we have to make sure that we know what type they have and that we know what type the donor is donating. And we do that, and you guys will do this in lab if you haven't done it already. You'll do this in lab. We're going to give you synthetic blood and some synthetic antibodies, and you're going to mix them together and find out if agglutination reaction occurs. So what is an agglutination reaction? Well, if it agglutinates, that means it's going to end up with these little teeny tiny sticky sections that are going to stick together, right? So you're going to end up with little aggregates inside. All right, so what does that look like? So here's untreated blood for everybody. So before the treatment, everybody is going to look the same. So you're not going to be able to tell the difference between A, B, AB, or O blood. But if you treat it with the antisera, you'll be able to see, right? So if you treat it with anti-A antisera, you're going to see an agglutination reaction. If you see the agglutination reaction, that means you know that they have those sugars on the red blood cells because you just added the antibodies. So if you add the antibodies and you see the reaction, you've got the sugars on the red blood cells. That means this is A because we saw a reaction with anti-A but not with anti-B. This one, on the other hand, no reaction with anti-A, but reaction with anti-B, that means it's B. This guy reacted to both A and B, therefore is AB. And this one reacted to neither, and therefore is O. So this is what you're going to be doing in laboratory as well. We're also going to add in another set of antibodies that you're going to add that are going to be the RH factors that'll give you that positive or negative that's going to add to that. So it's either going to be A positive, A negative, B positive, B negative, AB positive, AB negative, O positive, or O negative. And we'll go over that more when we get to the lab itself. Okay, so now we're going to talk about that positive and negative that I just talked about, that RH factor. So during the birthing event, fetal blood is going to leak into the maternal circulation, right? So this doesn't happen until delivery or much later, and so for the very first pregnancy, this won't be a problem. So at birth, we're going to have a little bit of fetal blood leak into maternal circulation. So if the baby is RH positive and the mother is RH negative, that means she's never seen that RH before. Now she's going to have seen it and she's going to create antibodies to that RH factor. That means that now if she sees another RH positive baby, remember she's RH negative, so the antibodies don't hurt her, but the second RH positive fetus can cause the fetus's red blood cells to agglutinate and then hemolyse, which means congregate and then break open. And so this can be a serious problem if we have a mother who is negative, who is dating a man or has had a child already with a man who is RH positive. Because that means that he likely donated an RH positive sperm to that embryo, which means the embryo is very possibly RH positive, and if it's RH positive and she has antibodies against RH, she's going to attack it. So this is one of the things they test for very early on in fetal development to make sure that they can give this woman some sort of immunosuppressants that are going to stop the creation of these antibodies. So I'm going to show you what this looks like. So she's RH negative, right? The fetus is RH positive, but the RH antigens aren't going to make it across because the placenta is going to have a barrier, so we're not going to actually have a problem until birth when we're going to end up with a rupture of this placenta and it's going to release that into maternal circulation. So then she's going to see those RH antigens. When she sees them, she's going to create anti-RH antibodies, and then during that second pregnancy, these antibodies are already in maternal circulation. They can go through the umbilical cord, get into the fetus and attack, which can cause hemolysis, agglutination and hemolysis, which can cause a miscarriage event. So what would we do? Actually, we would take this individual and we would make sure that she was given an immune drug that would help stop the creation of these anti-RH antibodies and keep them suppressed during the entirety of the pregnancy so that it wouldn't affect that fetus. So there is a specific drug that we can use to intervene with this process. Okay, so we talked about blood being responsible for carrying oxygen to the tissues, right? It carries oxygen there, it carries carbon dioxide back, and we can have a problem in that's called sickle cell disease. And sickle cell anemia is actually a type of genetic anemia whereby we make an oddly shaped blood cell which has a reduced oxygen carrying capacity. So the red blood cells of individuals with this disease have hemoglobin S instead of the regular hemoglobin, and that makes an odd sickle cell shape once it releases its oxygen. So it looks normal when it is holding oxygen and sickled when it has released oxygen. This is what a normal red blood cell looks like, just to kind of reorient you. Again, it's a circle like a torus with a little dimple in the middle. This is what a sickled red blood cell looks like. It can carry some oxygen, but not as much. And this is what's called cretinated, which is kind of like falling apart upon itself. So that's not a normal blood cell. What happened? Is that it? Okay, well, that's going to be the end of our lecture, you guys. That talks about different homeostatic imbalances. Let me just touch on this for 1 second before we go. I know I've had you for a little bit too long here, but the importance of why we brought up sickle cell anemia is because an individual who has 2 copies of the sickle cell gene is going to end up dying of sickle cell very early on, and an individual who has only 1 copy of the sickle cell gene is going to be able to carry oxygen, but not as much oxygen as a normal individual. Which sounds like it would be a problem, except in areas that are laden with malaria, it actually gives them a resistance to malaria. So we find that sickle cell anemia has a higher prevalence in African American descent, really African descent, because in areas of Africa we see high prevalence of malaria, and so having the sickle cell anemia actually confers a resistance to malaria, so it ends up persisting in those populations even though it would normally be weeded out. So, all right, that brings us to the end of our lecture. I appreciate you sticking with me this whole time. I will talk to you guys next time. Aloha.